Hello, this is chapter 13, Control of Microbial Growth. So this is basically a chapter on how different chemicals and agents work that kill bacteria in the environment. As we know, microbes are everywhere, and I had circled this term CFUs, right? So they did a study on this car and reminding you that CFU stands for colony forming units, which is down here, because we cannot be sure how many bacteria actually cause or form the colony when you grow them in the lab. So we want to use that term instead of, like, for example, cells, right? You don't want to use if there's for a million cells, there's going to be 4 million colony forming units. Anyhow, even a clean car like this has quite a few colony forming units sampled from its area, and I'm afraid to say my car is much dirtier than this. So I'm sure the microbes in my car are more plentiful. In order for scientists to work with microbes, there are precautions you need to take, as we do in our lab. And this is a biosafety level. This is showing number 4, which is the highest, which works with really dangerous viruses like Ebola, and where you need your own air circulatory system. You need to wear a head-to-toe suit so you're not exposed to anything in the air in the lab. So this is the most advanced, or the highest biosafety level. Here is a chart of the different biosafety levels. So it's from 1 to 4. And in our lab, we basically are mostly BSL-1, and there's some labs where we work with Staphylococcus aureus, so it'll be a BSL-2. But we're still practicing safe procedures, right? In BSL-1, they are microbes that, they don't really cause disease, minimal risk. You really have to try to get these microbes into a cut or something in order to cause an infection. An example of this is non-pathogenic strains of E. coli. Non-pathogenic obviously means that they're not going to cause disease. In BSL level 2s, we have basically varying strengths of disease-causing microbes, and they pose a moderate risk, meaning that, yes, Staphylococcus aureus can cause infection. We do take precautions not to come in contact with it. During BSL-2 labs, we do wear gloves. But it's still relatively low risk, because you're not going to, unless you sort of get a large inoculum into your body, you're not going to get an infection. Then we get into the more severe, dangerous microbes that are going to cause either serious disease, potentially lethal diseases, and you can even get them through respiratory transmission. And this would be something like Mycobacterium tuberculosis. And then what we saw in the picture before, we see this head to toe, completely isolating yourself from the environment. And these are going to be really high risk of aerosol transmitted infections, which are basically in the air. They can be fatal. So very few labs in the world actually are like this. And if you think about, like, Ebola is a really scary virus that has had some scares in New York recently, and if you did catch Ebola, the death rate is actually very high. So these are the different safety levels. Again, we are going to be working mostly at 1 and sometimes at 2, but we will wear gloves and practice aseptic techniques. This is just a fun little quiz if you want to pause the lecture and see if you can match them correctly. I'll go ahead and let you pause now. And we can go through now and look at the answers. So when something is sterilized, that means that everything that can cause disease has been killed. So that would be letter C here, where you're killing all forms of life, including, which is very important, endospores, and inactivating prions. As you know, remember prions, we studied them. They are proteins that can cause your own proteins to misfold and cause neurological symptoms. But prions are pretty rare to come across, right? They're in special circumstances. So if we think about, like, everyday life, the most common kind of most dangerous and hardest to kill would be the bacterial endospores. So basically, if you're in everyday life and everyday practice, if an agent can kill endospores, it's considered a sterilant. You can actually sterilize something and remove every single form of life that would be able to contaminate it. Well, what is disinfection is one, and what is antisepsis, right? So we have a small difference here. So for letter A, killing of microbes on living tissue, that would be your antiseptics. Okay, so if you think about what you put on yourself, hydrogen peroxide, for example, is an antiseptic. Alcohol is an antiseptic. But when you talk about disinfection, you can actually have an overlap of a disinfecting agent and an antiseptic, such as alcohol. But when you say disinfect, you're actually talking about inanimate objects. Inanimate objects like eating utensils, like the countertop, right? So anything that is not living, you would use a disinfectant on. For pasteurization, you should be sort of familiar with that with probably milk, right? So this is going to be your reducing of number of microbes in, this is supposed to be liquids, for consumption, especially targeting human pathogens. Okay, so pasteurization is something that you can only pasteurize liquid items, like juices can be pasteurized, milk can be pasteurized. Degerming is going to be what the nurse would do to you right before they give you a shot. They would take that small alcohol prep pad and they would like rub that area of skin. So that's going to be our mechanically removing the microbes from a small area. And then lastly, sanitization is going to be reducing the amount of microbes. Basically, a lot of these in restaurants, right? Acceptable levels by health officials. So this is basically the, a chart that you would want to be familiar with for this upcoming test, right? And so I want to show you that the first little section here is for use on fomites. Fomites, this word, is inanimate objects that, you could pick up a microbe from and it could cause you to get sick. So when you disinfect, you reduce and destroy the microbial load on inanimate items. You can use heat or chemicals depending on the nature of the item, right? So if you have plastic, for example, maybe the plastic would melt in heat. So instead of heat, you would be using like sort of a diluted bleach, perhaps. And then very commonly, right, we disinfect all kinds of stuff in our house. And then common things we use would be chlorine bleach, phenols. This is something that you find in Lysol. And then something called glutaldehyde. This is more severe. This is something that we'd find in hospitals. The word sanitization is also inanimate objects. So basically the difference is this is more rigorous. When you disinfect, it is thought to be more vigorous, more killing than when you sanitize something. Sanitizing is just kind of a lighter form of that. So when you clean your dishes, you're sanitizing them. Public restrooms are sanitized. And these are things, that you can buy at the grocery store, right? Any kind of detergent based on phosphates. And then there's something called QUATs. We'll talk about this as well in the chapter. Sterilization, again, when you say something is sterile, it is absolutely free of everything, including endospores, right? So all vegetative cells refers to bacteria cells that are alive. Endospores, those cells that can make spores, and viruses from inanimate objects. So sterile is like pregnancy. I like to say, like, you either are pregnant or you're not. You're never a little bit pregnant or you're never a little bit sterile. You're either sterile or you're not. And this is going to be, a very rigorous process, and it's for surgical equipment and things that you inject into the body. And there's different ways to sterilize. There is the autoclave that you guys have read about before. Different chemicals are sterilants, and radiation can be a sterilant. Down here in this area, we have living tissues, right? So this is where we have our antiseptics and we have our degerming. So not everything's going to die at the same rate. So there are some things to keep in mind when you think about killing microbes, and that it is the number of microbes matters. The more there are, the longer it takes to eliminate the entire population. The environment, what is with the microbes? Is it potentially some organic matter? Maybe it's in food, maybe it's vomit or blood. If it's protected by a protein or carbohydrates, right, they're going to have a higher survival rate. So perhaps you use the agent a lot longer, or you use a higher concentration. Temperatures, we learned that at low temperatures, everything slows down. Molecular motion slows down. So it'll slow down metabolism, it'll slow down the chemical work that the agents have. So low temperatures will actually kind of stall and slow the killing process, and even disinfectants, right? Because they're molecules too. And then the pH. So the pH will oftentimes matter when it's coming to the disinfectant. Sometimes disinfectants work best at like their pH, and if there's another agent there that changes the pH, it might not work as well. Then there's the concentration, right? How concentrated is the solution you're working with? And then you think about the dose, the intensity, length of time. Even radiation, sometimes, you need longer times with endospores than you do with like vegetative cells. And then the type of microbe does have a role in what or how it's killed, right? So different microbes, gram-positive, gram-negative, will react differently to disinfectants and antiseptics because if you remember, the gram-negative cell wall has that outer membrane, which is that protective semi-permeable layer. They have two of them, so they're kind of extra protected in that way. And then gram-positives are protected because they have an extra thick cell wall. So for things like lysing the cell, breaking it open, they might be a little bit more better at surviving that. All right, so do all control methods kill? No. It turns out that if an agent kills the microbe, then you want to say side, right? So bactericides, virucides, fungicides are all agents that would kill. Then there are agents that don't actually kill the cell or the virus, but they do stop it from reproducing. And those are going to end in the word static. So bacteriostatic, fungistatic, will inhibit the growth of fungi. So it's basically just stopping their growth. And then when you use something that's bacteriostatic, then what you're hoping for is that the person's immune system, right, if it's a cut on the skin, that the immune system will come in and then be able to clear the infection. Even though the bacteria is still alive, they're not going to grow. So your immune system has an upper leg, let's say, right? An upper hand. I don't know if upper leg's a thing. Okay, the death curve. So this is called the microbial death curve, and it's logarithmic. So when they do the log of it, it's a straight line. There's one term you want to know from here, which is the d-value. And the d-value is basically how much time does it take to kill about 90% of the population. So you're basically reducing that population to 10% of what it was. And then the time that it takes to do that is figured out, right? So every agent is going to have a different d-value. Every microbe out there will have a different d-value for how it responds to the disinfectant or the antibiotic, or sorry, we're not doing antibiotics right now, but disinfectants or antiseptics. And so you can see it's basically just if you had, this 10-fold or 90-fold killing each time and 10% survive, right? You can calculate how long do you have to expose that bacteria to in order to reach basically zero cells or reduce the population to the end. So the d-value would be that time here, right, to here. So in this chart, what's our d-value? Our d-value is 5 minutes, right? So 20 to 25, we've taken care of 90% of the population. Another 5 minutes will take care of another 90% of the population. So you go all the way until you hit 1. And the first part of this lecture, we're going to talk about physical control of microbes, and like physical control would be like temperature and radiation. We're not going to talk about chemicals. So what physically can you do to microbes to kill them? And the most common thing is to heat them up, right? So if we think about in everyday life, if there's something they're not sure about, there's water that you're not sure about, you would boil it. We know that sort of hot or heat can kill naturally, and this is because we know that heat can denature proteins. It denatures enzymes. It causes those proteins to unfold, become useless, right? Sometimes they can coagulate. So boiling is very, very common, right? I found this on the EPA website. So so we live in California, there might be some kind of natural disaster where if you don't have enough bottled water and you have a water source, you might want to disinfect it. So what do? You can boil it. And from the EPA website, boiling is sufficient to kill pathogenic bacteria, viruses, protozoa. If the water's cloudy, let it settle and then just take the clear part of it. It says a rolling boil for 1 minute should basically kill everything you need to kill in order to have it safe to drink. But they want you to boil it for 3 minutes just for extra padding, right? And there are always going to be exceptions. There's some microbes that can survive really long periods of boiling, such as hepatitis virus, and endospores are very hardy. But that is the best bet that you can use for water. Moist heat also includes the autoclave and also pasteurization. So let's take a look at those two. So the autoclave is a device that every single lab that works with microbes, or a medical office or clinic that needs to sterilize anything, will have an autoclave. It's basically kind of a big pressure cooker. It uses high steam heat and pressure to sterilize, and it reaches a temperature of 121 degrees Celsius, 15 pounds of pressure per square inch. And that's kind of a lot. So think about if you make a little inch on your body, right? An inch of whatever body part. And then you imagine like a 15-pound weight on that inch. And then you have, your whole body, every inch of your body with 15 pounds on it. That's quite a lot of pressure. So this is a sterilant. Autoclaves are thought to sterilize because they do kill endospores. They are killed by the high temperature, not really the pressure. The autoclaves are very useful for a myriad of things, right? Sterilizing surgical equipment, sterilizing, things that you use in the lab. And then, if it's a liquid or a broth, for example, the nutrient broth we use in lab is sterilized with an autoclave. But what might be the drawbacks of an autoclave is like not everything can withstand that high heat and high pressure. So if you have a kind of plastic that would melt completely, right, you can't autoclave that. So you can't sterilize everything. If you have, for example, vaccines or a culture of any living thing, you want to, no. The culture of living things would be dead. But if you had a vaccine, for example, and you wanted to sterilize the vaccine, it's going to be ruined in the autoclave. So what are some other methods that we can use to like sterilize, medicines that we would have to use? So you can't sterilize everything with an autoclave, but it is very functional for destroying microbes. And this is where all of your samples go after we use them in lab. We have a technician that puts them into an autoclave and then just throws them away because they're sterile. You have seen this. How do you know your autoclave is actually working? There are ways, right? So they make a special tape that is clear when it's before autoclaving, and then it turns black. You have black stripes on it after autoclaving. This is how you know that the autoclave has reached 121 degrees Celsius. If you autoclave something and the tape does not come out black, then you know that there's an issue with the autoclave and you want to get it serviced. All right, pasteurization is what we hear that milk and juices are pasteurized. So it is used in the food industry. We are targeting with pasteurization really specific microbes that are going to cause food spoilage and illness. And basically what happens is you're exposing the milk to a very high temperature, but for a very short time. Because if you start to leave milk, let's say at a high temperature for a long time, you're going to start to change the nature of the milk. You're starting to cook the proteins in the milk. So it's a short time not to spoil or change the nature of the food. So there's two kinds of pasteurization. One is called high temperature short time, which is when you see the word pasteurized, it's 72 degrees Celsius, but for about 15 seconds, and that's enough to kill the microbes we need to kill and not spoil the milk. Does it kill everything? Can we call that milk sterile? No. Okay, but it kills enough. It puts the microbial load down enough, right, that we can drink it without any issues. And this is also why milk goes bad after a while, because there are microbes in there. They're just at very low levels. And then there's the ultra high temperature UHT treatments, which is why you can actually have some milks in a box on a shelf at room temperature, right? So it's a different kind of pasteurization. It kills more microbes, and sterilizing temps can be reached with this. So that's why you can have a shelf-stable beverage with UHT. And then these guys here, right? If you go to a diner and you see these creamer guys, those have been pasteurized with UHT. So they can sit on the table and they're fine. Here is just a look at the pasteurization process, right? Again, we have a really large, it's a big industry, right? So we have large machinery. We can have high temperature, short time, ultra high temperature, and then this guy can go, without refrigeration. And then what we're looking for, you do not need to memorize these milk-borne organisms, but we can see that E. coli can be found in milk. This particular strain of E. coli is really nasty, and it can kill people. Mycobacterium tuberculosis, you guys know that one. There's Campylobacter jejuni, basically can cause diarrhea. Coxiella burnetii, Listeria monocytogenes. This guy right here, Listeria, if you're pregnant, it can actually cross through your placenta and infect the baby. So women, if you're pregnant, you should never drink milk or juices that are not pasteurized because of this threat. Then we have all other kinds of nice microbes, Salmonella, Yersinia. So think about how pasteurization has saved so many people from getting sick. And then you have the product out there, which is raw milk, right? So raw milk is not pasteurized milk, which means that it goes from the animal into the container without getting any of these guys, these potentially very pathogenic microbes. They're not killed. If they're in that milk, they're not killed, and it goes into the grocery store and then you would drink it. So I would highly recommend not buying raw milk because it is a risk to your health. And this is another kind of nice look at the HTST machinery that goes into to pasteurizing milk. All right, and another word here is that I just want to make sure you guys remember that Louis Pasteur, right, is the inventor of this process, this pasteurization process, where you expose a liquid to a high temperature for a short time and it kills most microbes that you would need to kill. So not to spoil the nature of the liquid, but keep the microbes down. All right, so now we're on to a second topic, which is dry heat. So dry heat is going to be incineration, one form of dry heat. And you know this, we just burn it, right? So we're going to incinerate or burn our, inoculating loops, inoculating needles. And so flaming is a form of dry heat sterilization. So sterilization, obviously, you're going to kill endospores if they're on that loop because you're flaming them, right? You're burning them. This guy here is something that you can also find in lab. This is like in the future, you might see these guys. This is an electric, it's kind of like a toaster, right? It's an electric coil. You would put your loop inside here and it gets very, very hot. So it's the same process, but instead of a flame, you would have this device. All right, I think that's it. All right, so dry heat sterilization, we're going to talk about incineration. The next one is going to be freezing. Now, freezing is completely different. Freezing, there's a couple of kinds of freezing. One kind is to kill or preserve, and the other kind is strictly to preserve. So let me just explain. So as you know, you can freeze your eggs, right? If a woman wants to get pregnant later on in life, but she's afraid she might, run out of eggs, or by the time she wants to get pregnant, the eggs will be old, so she can take them out and freeze them. Well, how do you do that, right? Freezers, if it's that kind of freezing, then it's going to be liquid nitrogen. Liquid nitrogen freezes instantly, basically. And what you need to know about freezing is that when water freezes, ice crystals will form, right? And water expands as it forms ice. So if you let water freeze slowly, there's more expansion there's bigger ice crystals that form. If you freeze something immediately in liquid nitrogen, for example, there's not enough time for that water molecule to expand so that the ice crystals don't really form in any way, and you can preserve that cell so it's intact. That's the benefit from liquid nitrogen. So that freezing, when you take the cells out and thaw them, there should be no change to the cell, the nature of the cell. Everything should be fine. All the molecules are there. All the membrane hasn't been broken or anything. So you can revive the cells, like you revive eggs for fertilization. An ultra low freezer maintains temperature at or below -70 degrees. And this is very low because your freezer at home is about -18 or -20. So that's also an ultra low. But if you want to 100% know that you want those cells back and you want those cells to be living, then you want to do liquid nitrogen because it freezes instantly and ice crystals do not form. All right, so this is how freezing can actually kill cells, and this would be a slow or fast freezing. But it's, so not liquid nitrogen. So what happens is when you do cool down a cell, right? So these little snowflakes, what happens is the inside will start to form ice, right? The water, which is the cytoplasm is mostly water, will start to form ice. And then the ice will break open. So if you see the extracellular crystallization, the cells will shrink. That's the slow freezing. So you do change the nature of the cell. You can kill the cell this way. And then intracellular crystallization, the ice crystals will pierce the cells. So when the ice forms on the inside with some quick freezing, those ice crystals will basically puncture the cells, right? And then basically like break it open. So think about when you put, like if you're impatient and you're like, oh, I want some cold soda right now, and you put a can of soda in the freezer and it, you take it out later because you forgot about it and it's, exploded on you. This is the same thing. All that water in the soda expanded and it punctured your can, right? So think about what would happen to the plasma membrane or cell wall of a bacteria. So basically like puncturing a balloon. All right, so for pressure is also used to control microbes. The sort of thing we're looking for here is going to be Clostridium botulinum. Clostridium is a spore former, as you can see here. The clear little areas at the tip of our rods, those are the forming spores. If you look around, we also see some free spores, right? Some spores where the vegetative cell is completely gone. And this guy, so I do want you to know the word, this species, Clostridium botulinum. It causes botulism. Botulism, we'll talk about the nature of botulism later, but it can kill you. It is sort of opposite of Botox, where Botox freezes your muscles, but botulism will cause your muscles to be unable to contract. And so, the most dangerous muscle we're talking about is the diaphragm, your breathing muscle. So your breathing muscle, you can't contract it, therefore you can't breathe. So Clostridium, let me just do a quick, it is a spore former. Most spore formers are being found in the environment because if you think about the environment, it's very topsy-turvy. It can be hot one day, cold another day. It can, be without rain for a very long time. So nutrients, moisture, temperature, all vary widely day to day. And so in order to survive those like wild fluctuations in the environment, the bacteria that have evolved to be in the soil form spores to survive. And so when you can foods and can vegetables, the spores of Clostridium are probably in or on those vegetables somewhere. Okay, so you can wash them, but you're not going to sterilize your food before you can them. So what happens is you put them in the can and then you put them in a pressure canner. Okay, pressure canner, because the endospores can be killed. You can boil the foods, right? But Clostridium botulinum can actually survive temperatures above boiling. So even boiling like a fresh food won't kill those endospores. But if you can them and you want to save them for later, you want to use the pressure canner. So that's all we'll say about Clostridium botulinum, that the endospores need to be killed by pressure. Another bacteria I want you to remember is Clostridium, and this is going to be Clostridium perfringens. Oh, I don't have the perfringens, but Clostridium. So Clostridium here, Clostridium here. What I want you to remember about Clostridium, it is anaerobic. Okay, anaerobic. So it is a bacterium that does not want to grow out in the open, but it loves sealed areas with little oxygen. So guess what a can is? It's a sealed jar with very little oxygen, right? So this is where Clostridium would want to grow. If you happen to get an infection with a Clostridium, with the bacteria from the Clostridium genus, it likes to grow in areas of your basically body that is wounded and does not have a good blood supply, because your blood is bringing oxygen there, and Clostridium does not like oxygen. So it'll grow in things like puncture wounds, in like septic abortions, where obviously like you're not going to a proper area, you're getting sort of I think I call it like alley abortion, but any kind of injury to your tissues that cause a block in blood flow, like a puncture wound is a great example, where oxygen is very low, and you have the Clostridium spore injected in there, then that's going to be a perfect storm for this kind of infection. So what they've done is they have this really great hyperbaric oxygen therapy. So hyper means more, baric is pressure, oxygen. And so for oxygen sensitive microbes or anaerobic bacteria, if you expose them to a higher than normal oxygen content, then they'll die. And oxygen causes a lot of free radicals to form inside their cells. They don't have a natural catalase system. They don't have enzymes to get rid of free radicals because they are naturally anaerobes. So they don't have oxygen as like a day-to-day item to deal with. They don't have reactive oxygen species that form on a day-to-day basis. So they don't have the enzymes to clear those toxic free radicals. So that's why hyperbaric oxygen therapy is good to use against these anaerobes. This is a look at Clostridium perfringens. Oh, here's the word, right? So this is our species. Clostridium perfringens causes gas gangrene. So the typical look is that you would have an area that's filled with gas, because what the bacteria is doing is they're fermenting the nutrients in your body. And with fermentation, they're producing gases as a waste product. So the gases will build up in your tissues and you'll see this kind of sac of gas. So that's a hallmark of Clostridium perfringens. All right, moving on to desiccation. This is drying. So we're just removing the water from these cells. So water, super important for chemical reactions to occur, right? Everything if you remember back to dehydration and like dehydration reactions and or condensation reactions, and then the hydrolytic reactions, those chemical reactions need water. And so if you take away that water, you cannot build up or break down anything in the cell. So if you remove the water, then we are going to kill the cell. And we can do it many ways. And a lot of this is with food practice, like preserving food. So you can air dry or smoke, which is our picture here, right? So if you have a constant fire going, we have a warm area, and we're basically just drying out all the water from in the meat tissue, right? The skeletal muscle tissue, and it can be preserved that way. Osmotic pressure is what you see here. Okay, so osmotic pressure is using salt. So this is going to be cured salmon. You use salt, and then jams. Actually, I should put the word jam here. Any kind of jam is also like this. Anything that's put into a high sugar or high salt concentration, it's designed to pull water out of those cells, right, through osmosis, and dries out those cell interiors, right, pulls all the water out. And therefore, if there are microbes on this salmon, the microbes would shrink and die, right? So remember that word plasmolysis? Plasmolysis, where plasmo, and it's like lysis. That is where the cytoplasm shrinks inside of bacterium, right? And it pulls away from the cell wall. So we have bacteria, and usually the cell membrane's right against the wall. But in plasmolysis, you'll find that, it'll still be attached to the wall in some areas like this, but the cytoplasm has now shrunken and shriveled to the center here, and that will definitely kill the cell. Then we have this picture. I'll just talk about this. This is just air drying, right? So these women in Pakistan are picking like apricots, and they're drying them just through the atmosphere, just taking the water away. And their last topic here is lyophilization, which is also called freeze drying. And freeze drying is different than the rest because air drying and using osmotic pressure like jams, like salts and sugars, those are aimed to kill the microbe. They are aimed to desiccate, the tissue and also, the microbes so they don't exist, and preserve. For lyophilization, this is freeze drying to preserve something like medicines or to preserve cells. So a lot of times when you order E. coli from the lab, from a company because we want to use it for a microbiology lab, the E. coli comes freeze dried. And then all you need to do is put water in it, and those cells will come back to life. So this freeze drying process does not harm the nature of the cell, and you can actually revive those cells adding water. Okay, so here are different images, right? So again, osmotic pressure, we talked about this, where we have a lot of solute molecules outside the cell. This was the cured salmon example, and it just pulls the water out of the cells, creating, if it's a bacterial cell, plasmolysis. Some foods are dried directly, right? Just like leave them out or use like your oven, just low heat for a long time. So raisins or jerky. And then other times, right? We talked about jams. That can also be, you can kill the microbes using that osmotic pressure. All right, I think this is one of the last physical ways to kill bacteria is using radiation. Now, there's two kinds of radiation. There is the non-ionizing radiation, which is less harmful, less energy. What non-ionizing radiation does, now this is that's in UV. So if you think about non-ionizing radiation, it's when you go out into the sun has this UV. We know that we don't want to be in the sun unprotected for a long amount of time because it can cause cancer. But what is cancer? Cancer is a disease of DNA mutations, DNA breaks, DNA, little damage to DNA that builds up over time. So this is so what UV does is make something called a thymine dimer, where the two thymines on DNA across from each other, they should be hydrogen bonded to each other, but the UV will come in, break those hydrogen bonds, and the two thymines will kind of hydrogen bond together like this, and then like this. And those are called thymine dimers, and they form due to UV. So think about the whole process of DNA replication. When a bacterium wants to divide, you have to copy the DNA exactly, right? If you have thymine dimers in there and they build up to so many, like such high levels, you're not going to get an accurate replication of that DNA, and you will basically kill the cell from making so many changes or making so many thymine dimers in their DNA. This is a picture of germicidal lamps that are used to emit UV. They're used a lot in labs to basically sterilize equipment, to get rid of any microbes that might be present without the use of chemicals. This is a nice example of a germicidal lamp in an air duct. So this can be something like if you are a tuberculosis patient, you have your own air circulation system. The air comes out of your room. Before it enters anywhere else, right? It moves through a duct, and the duct has that lamp in it to kill any mycobacterium that might be in your aerosol, in your droplets when you cough. It'll kill the bacteria within the duct. The second kind of radiation is called ionizing radiation. This is a higher energy. This kind of radiation is bad for us to be around. And they use this because it's a really good way to preserve foods, for example, that cannot be heated. It can't be dry. You don't want it dried, right? So how can we irradiate, how can we, for example, kill microbes on strawberries? I don't know what these are, but these are, I'm not sure, tomatoes? But anyways, foods that are exposed to gamma radiation move through a conveyor belt, and it's gamma. Okay, so gamma irradiated foods must be clearly labeled, and this is the symbol for gamma irradiated foods. This is known as the Radura. But it's not, so I think people are a little scared of the word irradiation or radiation because they think if they eat the food that, somehow that radiation is going to be passed on to you, but it is absolutely not, right? It's just this powerful energy that goes through the food, and it basically nicks the DNA and causes, the backbone to break on different, on the DNA and kills the microbes. If it nicks, and it'll nick the DNA of your strawberry, but who cares? Because the strawberry is going to be eaten, right? It's like, it doesn't matter that the DNA in the strawberry has some breakages in the backbone. You're not going to incorporate that DNA. You're going to break it down. So it's a way to safely preserve foods without chemicals, without heat. So ionizing radiation. I thought I had a picture. Okay. Okay, still going on here. So filters, we know how filters work, right? They move air through the filter, and the filter will trap the particles within the material. And there are going to be holes of certain sizes in the membrane, depending on how you want like what you're filtering. For example, are you going to be filtering viruses? Then those tiny holes will have to be really small, so it traps the viruses in the membrane and it doesn't move through. So HEPA filters or HEPA filters will remove microbes, endospores, and viruses as air flows through them. And this is just a schematic. There's nothing you need to know here, but just like a schematic of how filters work. And the arrangement of the fibers are in every direction because you want to trap as many microbes as you can. Sonication is using sound. So if the sound wave is enough, it can actually cause breakages in the membrane. So ultrasound waves make really big changes in pressure within the cell. In the liquid, it leads to cavitation, which is basically air bubbles, right? Big cavities of air bubbles. But air bubbles, like you can imagine them blow up. It causes a lot of pressure against the wall. It can disrupt cell structures and cause the bacteria to lyse or to collapse. So using sound is also a way to preserve different materials. Oh, sorry, this one came one slide too late. This is just another picture of filters. So if you wanted to filter liquids or filter any kind of medicine, right, that's a liquid, you can. So one thing to remember about filters is filters are used for both air, right? We can filter the air, and then we can also filter liquids, but we can't filter solids, right? So filters are great for air and liquids. All right, so we're at the end of part 1, physical control of microbe overview. So we talked about everything here. Okay, yeah, we talked about everything here. So this is a really great kind of synopsis of what we talked about. You want to basically look at mode of action and examples as the two major ones. You don't really, I think that, this is good to look at too, but let's know how it kills and some examples. All right, we talked about sonication, HEPA filters, and membrane filtration. All right, some questions. The absence of significant contamination is? So when we talk about the absence of significant contamination, we're not talking about sterile, right? Because we're just talking about significant. And we know that sepsis is dirty. Septic tanks are dirty. Asepsis and antisepsis, these are really close, right? But antisepsis is that's an agent, right? An antiseptic. So that's not the term you used. It is asepsis. So asepsis is the free of significant contamination. Bacteriostasis, remember that was when you when a chemical agent or a physical agent does not allow the bacteria to reproduce, but it keeps them alive, but they just can't reproduce. Which of the following best describes the pattern of microbial death? Okay, so the cells in population do die at a constant rate. Remember, that was the D value, and you can determine the D value by looking at how many cells die. You can determine the D value by determining when 90% of that population has died and only 10% survive. Okay, the time it takes to kill a microbe is going to be actually proportional to the number of microbes. So if you remember that chart, that graph we had, the time was on this axis, and then the number of microbes were here. So the more microbes you have, the longer, and we're going to always have this like linear curve. So that's going to be a longer time. If you only had like this many to start off with, this many number of bacteria, then the curve looks the same, but it would take a lot less time. Okay, so this is the answer. All right, this is part 2.